No?
It says the speaker is muted, so.
Okay, so now it works, right?
Is it better now? Yes, it is better.
Oh, okay. Okay, good. So the other aspect is the biomedical informatics. And for the biomedical informatics, we have been working on the electronic health records data and the patient data, like the epidemiology data. We have been using the AI and machine learning to do the disease progression prediction and to categorize the significant risk factors for the patients in clinical side. So that's the major two branches of my research. And we have demonstrated fruitful and high impact journal publications in these different branches and on using different type of data. And the major is to use the AI and machine learning to identify the research findings and share with the research communities. So from my research experience, using different type of data and on different research aspects, my experience and my feeling is right now the multi-modal data are really essential and critical for us to advance the research and also to advance our findings. So because the multi-modal data ranges from the medical image, the clinical notes, the tissue and the longitudinal EHR health records data and the genomics, omics, and spatial omics data, all those different type of data. That is what we call the multi-modal data are very essential because they bring in comprehensive insight. Because each of the data is only one layer of the disease. So the multi-modal data can give us more comprehensive insight of the disease. And it also, when we build the AI model, if you rely on only one layer of the data, sometimes it cannot be that robust or accurate. And so it also enhances our AI model accuracy and robustness. And this multi-modal data also enhance our mechanistic understanding from the bench side to the bedside, how the disease is progressed. And it will enhance the cross-cohort generalizability and lay the foundation for the next generation AI using this multi-modal data. So that is what my research experience that brings me to this layer of the multi-modal data. And regarding the multi-modal data, we have been using different type of data sources, and we have been using the national repositories that have a lot of data there and multi-modal data there. For example, the National Clinical Cohort Collaborative, there are a lot of social determinants and the electronic health records data, and the OFAS research and UK Biobank, one Florida. And there are also other cancer-specific database like the HubMap, HTAN, CapTag, and TCG. They are all the multi-modal data. They have the clinical data information and they have the omics information. So all those data resources are very valuable for us to look into specific disease or to identify specific biomarkers to handle the complex disease. And regarding the tools, we have been actively using the graph AI methodology. And since I think when I started my research career, and we have been using the graph convolutional network and the graph search, and more recently we have been using the graph transformer, and right now we have been using the graph transformer for different purposes, such as the transfer learning, domain adaptation, and more recently we have been using the graph enhanced large language models for us to leverage the data and to enhance our research studies. So since we have been using the graph AI as our methodology and our approach, and we have the good resource of multi-modal data. So I would like to showcase some example research studies of when graph AI meets multi-modal data in the area of bioinformatics and computational biology. And this is the first section of my research. And next, I would like to showcase the graph AI with multi-modal data in the area of biomedical informatics. So it is majorly divided into these two branches. So before I dive more specifically into a specific research study, I would like to introduce about the data in this topic, because you have to understand the data first before you understand what I'm doing. So you may heard about the omics word for a long time, because when you go to hospital, maybe your doctor will ask you to do the sequencing. Sometimes you can ask to do the genetic screening. to avoid special disease and for disease screening, sometimes like that. So you have heard this omics word, I guess. And since a long time ago, since 2006, the bulk omics technology have become available. The researchers can take the tissue biospecimen from the patient and do the omics. And so you get the omics profiles from the tissue. That is a bulk tissue, which consists of multiple cells inside. So that looks like this figure.
So what does omics mean? I don't know. What's the omics?
Omics is a general word that includes transcriptomics, genomics, transcriptomics, and proteomics. It's a general word. Of course, all those different layers of molecular sequencing. So we have the omics data that at that time available for patient tissue, bulk tissue, that consists of multiple cells. And more recently, the technology becomes more advanced, so people can measure the omics data from individual cells. And more recently, not only individual cells, we can take a slice from the tissue, that is the biospecimen, and we take a spatial slide, and then we can measure each individual spot on this spatial slide and get their omics data. And more recently, it is more advanced, is we get the single cell level omics data from the individual cells on this spatial slide, which is higher resolution. And the data is more advanced, the technology is more advanced. So that is the development of how omics technology becomes available during all those years. Okay. And next, I would like to showcase our research study based on this advanced single cell data, and especially single cell spatial data. And these are the three major companies. I'm not sure if you have heard about. They have provided the technology to researchers, and right now, if you look into the top Nature Cell Science paper, they all use this cool technology because it is the so-called most advanced technology right now to sequencing the individual cells. And the Cosmax technology provided by the NanoScreen company and Visgene and TENS. These are big companies that provide this high advanced technology. And this is an example of the real data. And this is a small spatial slide that take from a lung cancer patient. And on this spatial slide, individual dots represent an individual cell. Different colors represent different types of cells. And on this small slide, there are 83,000 cells detected, and each cell is measured with their gene expression data. That is what we call omics data.
So this one, like this on the, you say liver or lung?
Lung cancer. This is a lung cancer.
So what do they do? Take tissue and take a picture of that, or what?
Yeah, they use the image technology to measure the signals. What genes is expressing, is detecting the signals. They take the slides from the lung tissue. Yeah, lung tissue.
Lung tissue,
Yeah.
picture.
Yeah.
Just
Kind
high
of.
resolution picture.
Yeah, high resolution, super high resolution at the individual cell level. So that is the most advanced technology.
Microscope.
It is not it's more advanced than microscope. It's a more advanced technology. They call it as multi-channel staining.
And how big is
Fluorescent.
fluorescent?
Yeah,
microscope?
fluorescent. Yeah.
So how big is this image? Like how big?
It's very small.
Small.
I don't remember the exact level, but it's super small, so that they can measure the individual cells. So it's high
So we can
resolution.
see the cells.
Yeah.
But then how do you get the genes and so on?
So they will do the multi-channel fluorescence, so they can detect the signals of the genes for individual cells. So that how this technology develops, and so they get the data here. And as I mentioned, this is multi-modal data because they have the image, they have the multi-channel image, and they also have the cells, and they have the location of the cells, and they have the detected signals of each individual genes of each individual cells. So it is purely multi-modal data.
So what's the dimension, physical dimension of this, like I said, 10 to the 2 millimeters by 10 to the 2 millimeters, this what's the dimension?
So a cell is usually.
No, this whole image.
You mean,
Thousand cells.
I think it should be 10 to 20 micrometers. I would, if I don't remember
That's good.
the unit correctly. Yeah.
I have a question. So this is not the healthy, this is the part that they take from the patients after they are diagnosed, right? This is the cancer part.
Well, because for example, they have diagnosis this patient to belong tumor tissue, so that we can extract the biospecimen
Yes,
from the
exactly.
patient.
So in this case, this is already true positive, right? I guess you are going to, let's hear your results and I will ask.
Oh, I just introduced the data. I will introduce more results in the following slides. Yeah, it is. I just want to showcase what are the data that we are dealing with. And this is some other examples from different lung cancer patients. And you can clearly see that, for example, the first three is lung 5-1, -2, -3. They are three data from the same patient. And the lung 6, lung 9, and lung 12, they are from different patients. And from this technology, from the data that we have, we found that the data and this single cell spread data from different patients are very different. We should indicate that this patient microenvironment, tumor microenvironment is very different. And also highlight the critical needs of this technology, why we need so high resolution insights into the patient, because they are different. If we cannot get this high resolution insight, we cannot distinguish where the tumor cells are, because it is very complex. It's not purely tumor cells. It is infiltrated with other cells, and it's complex. And this is another example data from another major company, that is the Vision Scope. And they also have the similar technology, but the difference is their number of genes detected is different. And this is from a liver cancer patient. And they showcased their capability to do this single cell spatial data and showcase their power of the technology by releasing all those data from different cancer types. And this covers 8 cancer types, and totally they released 16 data sets and a lot of cells on this spatial slice. And from this different cancer, you can see that the different cancer, their different tumor microenvironment shows very different patterns, and which is very fascinating because we don't know this before. If we don't have this technology, we don't have this picture. So that's the example of the single cell spread data. So from the example, we identify that there are great data opportunity because it is multi-modal data at the single cell level, high resolution. We have the images of the cells. We can capture the morphologies of the cells, and we have the omics. That is, for example, the transcriptomics, the gene expression data for each individual cell. So it's multi-modal data. But it has very challenging issues as well. As I mentioned, the signals detected, because the technology is very advanced. So the signals detected is not sufficient. It's not as comprehensive as our bulk sequencing technology. So the data detected is not sufficient, and the gene coverage is not sufficient. So that's the issues of the technology. And if we face these issues, we have been working on this data using graph AI and multi-modal data to overcome the data challenges. That is the first study that we have been worked on, that is called Seagram model. This is a paper that published in Nature Communications in 2023. And at that time, this is the first, I think this is the first study that we have been handled this multi-modal data. And as introduced in the previous slides, we have the multi-channel images that are imaging the cell signals. You can capture the cells and their morphologies through these multi-channel images.
How many channels are there in this? How many channels?
So different technology from the 3 companies, they are slightly different, and mostly they have 3, at least 3. At least.
3 channels, right?
Yeah, at least.
Take a picture. So there's at least 3.
Right, at least. Some of them can expand to
Each is
the
got, how many bits, each pixel is how many bits, like 8 bits or something, more?
for each cell.
No, each, is an image. That's a
Yeah,
picture.
it's image.
So you have 3 channels
Yeah,
and you have 3 numbers at each pixel.
yes, that's right.
And what's the range of each pixel?
Yeah, that's a good question.
A regular image, we have 0 to 55, right? Of GPU.
Okay.
So here is, you know, how much you like, No?
I don't have a good estimation of that level.
Yeah, that's fine, but then
Uh-huh.
what's the size of the image?
The size of image, I think the whole slide image is, it depends on, so after we got the data, it depends on how much the spatial slide has been scanned. They have different protocols, they have their fixed size of how much spatial slide you can put into the machine. Yeah, and but here we actually have cropped the whole slide into small ones to as an example. But here, well, we have the multi-channel images and the gene expression and spatial location, this multi-modal data, and we convert all those
And these
multi-model.
genes thing that, so one is a picture, you take a picture of lung of
Yeah.
people die, right, with the tissue.
Right.
Now, what are these genes, where do you get this transcripts, the genes? Where do you get that data?
Yeah, so their technology can get the gene expression data simultaneously when they do the imaging.
So that's a different sensor. It's not the same as the imaging sensor.
There's other sensor which gives you genes or?
Yeah, I assume so because I'm not the protocol part. I'm not very familiar with how they get the technology work. But that is because when the machine scans the image, I won't see the scan image in the slides, visual slide, they will get the multi-channel images as well as gene expression data directly from the machine. So that is how we got the data.
Generally, you have 40 or 50 channels that the machine collects.
Yeah.
Those are called markers. And then there's a Boolean process where you combine the markers together. Each marker is a fluorescence on a particular gene set, whatever. And then you have a particular cell that light up in CD4 and CD8 and CD4. And you decide the Boolean combination of what type that cell that actually is. And that's all done by the machines generally. And so you'll get back either a set of markers or the gene allocations and stuff. So that's part of the new level of systems that we're getting now.
Well, we
Yes,
could,
yes, thank you.
Same size as a WSI, 100,000 by 100,000.
how accurate are they?
Accurate.
Identify genes?
Really that's still a state-of-the-art thing, but the commercial products are quite accurate. There's several open source solutions, Sequoia from Stanford, a couple of things like that, that will get you very accurate for the 500 genes and good approximations for like 10,000 and 20,000 genes.
Yes, thank you so much for the explanation.
I
It's
have
really
another-
expert.
I have another question. Do they miss some genes?
Yeah, of course, that's the issues that I have mentioned. We have a lot of issues with this advanced data. Yeah, because the technology keeps evolving. So we propose this work to handle the data issues. Because the signals detected sometimes it is not that accurate. So we hope to leverage the morphology images to make up for that issue, to enhance the signals of the gene expression so that we have less issues of the data. And the design is we will convert this multi-modal data into a single cell spatial graph. And the spatial graph is constructed based on the cell spatial locations. And each edge represents two neighboring cells, and each node represents a cell, and each cell is accompanied with its specific image and its specific gene expression. So in this way, we construct all those data into a single cell level spatial graph. And on top of that, we have the model architecture. We have the image-based graph transformer and the gene expression-based graph transformer, and we get the embeddings of the gene embedding and the image embedding. And we also concatenate these two embeddings into a hybrid embedding. And through here, we have decoders here and reconstruct the gene expression through these three embeddings and through the MSE loss, as shown is here. And after the training, the wild training of the model, we get the well-learned latent representation. And this latent representation and also the reconstructed gene expression. So the reconstructed gene expression learned from this model will be used as the enhanced gene expression data that can be very useful for us to do the downstream analysis and for us to uncover maybe some biological signals. So we didn't rely on the raw data, we have the enhanced data for you to use, which we have proved more accurate. And based on this latent representation, we could do the cell clustering, which help us to identify the spatial cell types from the spatial slides. Because after you get the single cell spatial data of the spatial slide, as I showcased before, you don't know which cell belongs to which cell type. So you still want to identify the cell types on the spatial slide. So we use this latent representation to do the cell clustering, which helps us to identify the cell types on the spatial slide. And
So in this one, so you're
Uh-huh.
saying you make a graph, right?
yeah,
And each node in the graph is the same.
cell, yeah,
Yes. And you have some the data you put in this, and you get this latent representation, you have three here, right?
yeah,
GMI, GHI,
GGI, yeah.
So each represent what is that?
So the GMI represent the head GMI represent reconstructed gene expression from the image purely based on the image profiles.
Okay, good.
And the GHI head represent the reconstructed gene expression from the hybrid embody of image and gene expression.
Okay,
Yeah, together. And this one is the reconstructed gene expression of based on the raw gene expression imbalance here. The ZGI is the imbalance of the gene expression data, and this is the reconstructed gene expression data.
so once you get all those three, then what you do with it? What,
So we have the loss constructed
yeah,
here, and then we train the overall model based on this loss.
so that's the way you train, right?
Yeah.
So you're done training.
Yeah,
So
that's
you
right.
get this representation, then what do you do?
So we will use we actually got two output derived from this model. One is this latent representation we will use to do the cell clustering, so that we know which cells belong to a specific cluster that belongs to a specific cell type. And we also get this GHI. This one is used as the reconstructed gene expression data. That is what we call as enhanced gene expression, because it merges the image information as well as the gene expression, so that we call it as enhanced gene expression data.
So when you cluster, why do you want to cluster? Once you cluster, what does it tell you? That these cells are similar? Or
Yeah, right, exactly. So it tells us what groups of cells are similar with each other, but different with the other group of cells. And these cells are more likely to be the same cell type, which is very informative, because when you got the cells data, you always want to tell where the cells are, right?
So once you get clustered, then how does it help the patient to, that, you know, what you do with them?
So is it the case that one cluster might be cancerous and the other one is not, or something like that?
Very likely. Yeah, I will showcase and I will have many other examples here. For example, we have the clustering, and this is the raw data. We can tell the cell types. For example, okay, the point that so for example, the different colors represent different cell types here. And the anticellular cell is typically more likely to be tumor cells. So in the tumor patient, and there are myeloid cells and the lymphocytes, endocellular fibroblasts, different cell types based on the latent representation. All those is based on the latent representation. So it helps us to recognize the cell types.
So what method you use to cluster? What's the method?
It is we use the latent clustering. We have several different clustering approach. Actually, based on this study, we use the class, I think.
M plus, right?
Amy?
Yeah, K-means doesn't work well, actually. We have tried K-means, but it doesn't.
M plus, they are.
Yeah.
So the main point is that you get one better representation.
Yeah.
The better representation helps you to cluster.
Yeah.
right?
Yeah.
And we could have used just raw data, cluster doesn't work. so-
Yeah, it is an example why our enhanced representation is better than the raw. And I also showcase another example. The below one, the lower panel is the enhanced data that we learned. It shows strong contrastiveness that indicate of the specific cell types compared with the raw data. Because if you look into the raw data, the signal is very poor. So you cannot tell which is which.
So what, I mean, what the is the graph structure is important in this one, like if you don't do graph, will this work or not? at all?
We do try. So that's why I always sell graph AI in my presentation, because we do try the graph AI indeed works, because we have incorporated the spatial location of the cells. It indeed matters, because cells in their different locations, it has meaning.
Yeah, we have a Q&A, expert in graph, right?
Yes, I mean, I also have some lung cancer, but can you go back to the previous slide? I want to ask the question. So, you talked about genes as nodes in the graph, and then you create one graph for each cell,
Yeah,
right? This is graph classification.
yeah,
So I heard node classification. So this model is trained on graph classification of cells.
yes, yes. The graph is the node of cells. That's right.
One question, so in this training model, this is an unsupervised training model, is that correct? Because you don't have to have ground truth, is that right?
I would say it is self-supervised because,
That's right.
yeah, self-supervised, yeah.
Yeah, you don't have to have.
No, we don't have
You
access.
you have the supervision right from the original data.
Yeah, that's
You
right.
just check.
Yeah. We don't have extra labels to help us learn that. It's purely based on the data. And so this is more, this is the first work I would like to say to handle this type of data. And next, I would like to showcase of the real, why we handle this data and what the translation of the impact of this data and what insights this data can help us in human health and in disease. So that inspires me to work on my favorite topic, which is the drug-related topic for drug discovery, drug repurposing, and biomarker discovery for the better therapeutic target. So that leads me to use this graph and multi-modal data and this advanced data to uncover the drug response. And also this, before I dive into more, I would like to give a little bit introduction of the cancer treatment. So right now there are different of cancer treatments available, including chemotherapy, immunotherapy, targeted therapy. There are different type of cancer treatment available right now. Even for a chemotherapy, there can be multiple drugs and different regimens and different treatment therapies. However, even though there are so many treatments, so many therapies, there are still patients that doesn't respond. That is what we call drug resistance. And it is a very critical issue. And there are a lot of challenges how we overcome the drug resistance. So to overcome that, we need to understand first, if the tumor cells in the tumor layer, they present different response to a specific drug. Or they are heterogeneous or homogeneous. So, and how tumor cells interact with the microenvironment to acquire the drug resistance. All the tumor cells, they just form a unified system to fight against the drug. So we don't know that. And if the tumor cell present heterogeneous response to drugs, whether this heterogeneity comes from its intrinsic molecular characteristics or it's from the extrinsic driving force to make it become resistant. So we don't know all those, but to solve the problem of drug resistance, we need to understand and scrutinize the physical constraints of tumor cells and to understand all those issues and to overcome drug resistance. So that's the background. And with this advanced data, which makes it become more and more available to solve these problems and to answer those questions. Because we have this advanced data, we can get into the inside of the tumor cell microenvironment, and we can understand different locations of the tumor cells, how they respond to a specific drug. So this technology is make everything become achievable. So we have proposed this work to link the data to real world impact is to understand the cell level drug response to drug to treatment. And
I think you can spend more time on this to understand. So you
Okay.
have genes, right?
yeah, we have
Cell lines, can you explain what are the needs?
So- Yeah, so the overall design is shown in this diagram is we have the drug knowledge base. We have, because the government have spent a lot of money to get the gene expression data of cell lines, and we have the drug screening profiles of all those cell lines. So we all have this knowledge. This is our knowledge. We want to transfer this knowledge to the single cell spiral data to learn whether a specific cell is respond to a specific drug in the single cell spiral data. And the goal of this model is to uncover the cell's response to drugs on the spatial slide so that we understand the tumor cells on the spatial slide, which tumor cells respond, which tumor cells are not. So the goal is to transfer the drug response knowledge to the new single cell spiral data.
So the tumor, people get tumor, they get sick and they die, right?
Yeah,
So a tumor has many cells.
yes, that's
And
right.
so you take a picture and you take some data of a tumor.
Yeah,
And then the cell lines is saying that there's some data that particular cell or particular drug will work and how well,
Right,
right?
yeah,
So, and then there's relationship of cell and genes. That's the first step, right?
the,
Cell lines and genes. Is that
Yes.
what it is?
yes, we have the structure data
Yeah.
and the rows are the cell lines,
Yeah.
different cell lines and different genes and different cell lines screened by different drugs.
So given all that, so if I'm a patient, then the research is saying that you can take a tumor, some data picture, and predict which drug will help to reduce the tumor. Is that the idea? What's a bigger idea?
Yeah, if we got a new patient, we want to understand whether it is respond to a specific treatment, we will get a slice and see how much proportion of the tumor cells will respond to a drug.
Mhm.
So that is something we can predict. If there are more tumor cells that can respond, that means these patients are more likely to respond to this drug treatment. So that's the
And in this also, again, you represent this as a graph and then you predict.
pardon.
I mean, in this problem also, you represent this data as a graph. And then
Oh,
predict.
yeah,
the
that's a good point. So that's the model architecture. So here we have we use the domain adaptation. So we have source domain and target domain. The source domain is the drug knowledge that is cell lines data. And so we also have graph over there. This graph is constructed based on the cell line similarity. And we also have the single cell spatial data because we have the spatial locations of the cells. So we have the spatial graph over here and also the gene expression data of each individual cells, the tumor cells here. So that is what we call source domain and target domain. Source domain contains the drug knowledge, drug response knowledge. And this model actually consists of three major components. And the first one is the feature extractor that extracts features from both source and target domain. And we learn the latent representation showing up here. And the second component is the domain discriminator. And the goal of the domain discriminator is to distinguish the source and target domain. And actually it is want to fuse the representations from the source and target domain so that it is mixed very well. Because that is our goal, because we want to use the drug response knowledge. That is the third component is the drug response predictor over there, which if the latent representation from the source and target domain can fuse very well, then we can use the knowledge that is what we call the labels from the source domain, and then to predict the labels of the
Just to
target domain.
clarify, the source domain is that the knowledge and
Yeah.
target domain is a particular patient.
Right,
Idea?
yeah.
So you have a lot of knowledge, you have sources,
right.
and then you want to transfer and say, well, this particular target patient, what can you do? Is that idea?
That's a general idea. We want to predict for specific patient and these tumor cells is more likely to similar to which cell line. You can think that in that way, because we want to fuse the source and target domain in the latent representation and mix them very well, so that if, for example, if the tumor cell are more likely to be similar with the drug resistant cell lines, then this patient may more likely to be drug resistant.
Because, AI is very standard thing, the terminology used of source and target domain, you have large data set, you train on that. And then you have particular hospital, particular set of patient, right?
Yeah.
So if you use that model for that will not work. So what you do the knowledge transfer,
Yeah,
take the source of this knowledge,
right,
adapt to the problem.
right.
Is that the same idea here? or-
Yeah, same idea. Yeah, same idea.
Good.
But can I say, so in machine learning, we use this, but in cancer studies, what they usually do is they have some patients, and when you, for example, are diagnosed with cancer, they take a sample from you, and then they try to identify some genes like ALK positive, for example. In the past, it was very binary. If they find ALK positive, you have a drug, you take the drug for all your life, right? Now they are trying to do non-binary, but something probabilistic. So from X patient, all patients, they know which drugs work on which patients, and then they have a new patient. They take a slice of the tumor from the new patient, and then they say which drug we should give to this new patient. So the old patients are the source domain. The target is the new patients, potentially multiple. So the idea is to find the drug that will keep them living alive. Because you take that drug, you survive, and then after some time, even that drug stops working. So they need to know which cells are causing the cancer to come back, so they will develop something new on top of it. So that's the idea. That's what I know.
I think maybe it's similar, but the source domain here is just I think the difference is the source domain here is we use the existing knowledge base. So that's maybe the difference a little
So
bit.
in this one, like you start with picture of tumor tissue, right? That's
Yeah.
what it is.
Right,
So you do all that, you come up with a single cell spatial drug responses.
yeah,
So you're saying that particular cell, what drug will respond, that's outcome.
right. We can tell on this tumor tissue, which tumor cells respond. And that's the result here I would like to showcase. And this is a lung cancer spatial slide and individual cells here, and the blue ones are the tumor cells. And it is surrounded by the other cell types. And this is our prediction on a typical lung cancer drug, which is cisplatin. We found that tumor cells, some of the tumor cells present sensitivity and some of the tumor cells present resistance. And moreover, we also tested another drug, and this result is shown, this is a microenvironment. We have detected different microenvironments surrounding sensitive and resistant cells, and this is an example. This is cisplatin lung cancer drug, and this is docetaxel, another lung cancer drug. And we found that for the same tumor cells, they present different response to different drugs. And very interesting is when we combine these two drugs, more tumor cells become sensitive, which is a good sign. Means that this combined therapy may be more effective than the single drug. And it is also proven in the clinical practice. So this serves as a real-world validation of our model, how it works and how it can be translated into clinical side.
Yes, that's the way you should have explained, right? Give motivation first to explain how, okay, why you are doing it. So, so you are saying like when you put a drug, so it goes to person, then it will impact particular cells.
Yeah.
Another drug will impact particular cells.
Right.
And then hopefully cure.
Yeah, and we also highlight that with this technology and this new data, we find that the tumor cells indeed present different response. Even when we always talk about the patient resistance, but we never goes into depth. Why this patient present resistance or sensitivity or sensitive? If this patient present resistant, it doesn't mean every tumor cell is resistant. It means some or major or certain subset of tumor cells present resistance, and this gives us in-depth insight of it happens. It is occurs everywhere. So
how is it currently done? Like, people get sick, so go to doctor, they take all these pictures and so on. So currently, they don't do this, people do it, the physician, they look at this kind of data or not really?
yeah, that's a very good question. So because this technology is very advanced, so maybe it is not possible for it to be adopted immediately in clinical practice, but there is always solutions. For example, we are working on some other work. I haven't have time to showcase here yet. We have developed a large language model to identify and to learn the omics data from the images purely, which means that if we can identify the omics data from the images directly, the text visual large language model, and then we can, as long as the patient can be digitalized pathology, as long as we get the digital pathology of this patient, we can have the solution to predict the cell level response to drugs. So we don't need this cool technology to sequence every patient. But if we can get the pathology image of this patient, it provides us the potential to look into the drug response. So that's some work we are working on. Haven't published yet, but we are working on that. So that makes the this more available in clinical practice, and because most hospitals, they already have digitalized their pathology, their patients. So so that is about this work. And because I have my favorite research topic on the drug sensitivity resistant discovery. So on top of my previous work, we also continue to work on this topic and using graph AI and the multimodal data. And here is this next is more recent work, and we have been using the graph-based large language models. And to use the large language model, we need a large pool of data. So before we can do that, we need a curated database and a comprehensive database so that we have enough data for us to train the large language models and to predict the drug sensitivity and the resistance.
So the reason we use large language model because what it has a lot of knowledge about drugs and cells and genomics and so on. Why do you use large language model?
Yeah, that's a good point. So the reason why why it inspires us to work on the large language model, because we want to unveil or to identify the potential of generalizability, because we want to build up a model that can predict every cell or almost every patient. We are not narrowed or limited by a specific deep learning model that have to be trained on small cohort of data, which cannot be generalizable to other contexts or disease. So that expands us to think about whether there is a model that can be generalizable enough and predict accurate response in different contexts. So that's why.
And the reason you can do it because large language model is a millions of these.
Right.
It is absorb all those things.
So it makes it easier to do generalizations.
Yeah, that's our motivation and inspired by that. So we have curated a lot of data, and that is more recent work. We have curated a lot of single cell data that have been treated by specific drug, and we get all those data, collected all those data from the public repositories, PubMed, Geo, and SRA, and different resources, and we have built this database. And we have also analyzed all those data, including the standard data analysis and also the cell notation advanced analysis and also the functional notation, which gives us a new paper on the database.
So this data is available publicly. You are using that. You haven't collected yourself.
No, we collected each lab's published data and build this database.
So like you have some instrument to collect this kind of data. What kind of?
So for example, a researcher who work on their specific topic on drug, they will generate some data, and then they will publish a paper, and then they will release their data to the public repository. But all those are scattered. No one think about this and collect all those treatment-related
So
data.
you're not necessarily getting all your data from UN. You're getting.
No, it's all public
that you
repository.
put
Yeah,
together
public.
on-
Yeah, we did the first time to collect all those relevant data together and analyze all the
So
stuff.
if you are a researcher, you want to collect your own data, what do you need? What kind of instrument? Equipment, collect data? With the big machine that fits in.
No, because all those data have been published, so we just
Suppose
uh-huh.
it's not public, like new data.
new data.
Yeah, how would you collect it? Like, what do you need?
We need to
Big
talk
machine?
with clinician first to get the biospecimen, and then we go to the machine to generate the data, and that's a lot of money.
How expensive is the machine?
I think for the single cell or the single cell sparrow, it can be a lot. I think for the single cell, it's $3,000 for some, yeah.
3,000 nothing.
And for the single cell spatial, it is 6,000
That's per run.
full run. Yeah.
Each listing of a patient.
Each,
I don't know about the machine, that's millions of dollars.
no, so that's what we, right?
Yeah.
Suppose we get you machine here, if you have the higher, so how much will it cost?
If we,
Ability is to go around the world and get data from there, not medium.
yeah.
50 or 60,000 euro, or maybe go to 100 grand,
We never,
Euros.
$1,000 dollars.
dollars.
We never dare to generate data ourselves. It's too expensive. So we collect, yeah, we build this comprehensive database, and all this database covers different treatments and also different cancer types. Different patient tissues, cell lines, CDS, PDS, and different treatment, chemotherapy, targeted therapy, immunotherapy, and the ranges across different cell types. And that's the database. And since then it is done by 2023 and published in 2024. We keep collecting all those data. Actually, we have version 2. Hopefully, it will publish soon. So on top of the database, we have built this graph enhanced large language model, and we want to predict the drug sensitivity and response across different contexts. So that's the motivation here. And in this one, we have the top expressed genes in the cells, and we have tokenized this and put into the transformer. And we have this graph, which is a pre-knowledge actually. And this is a purely knowledge graph. And we incorporate these two embeddings together and to predict the drug response. And this is it is not that hard, but it's just transformer blocks. So
So in any of this is from UF, and UF is a big medical school, right? So they don't collect their own data also.
yeah, that's a good point. Yeah, that's a good point. If we actually collect all those from public repository, we didn't talk to specific UF.
Data, when you have hospital.
No, if you want to get the UF specific data, it takes time because you have to negotiate with them, you have to communicate with them about different policies, how you use it, a lot of things going on. So
Most clinicians are not interested in really in the complexity of sharing their data.
yeah, sometimes they don't want to share.
It's the government sources that
Yeah.
will make the deals but the bigger hospitals will have internal use of their data
Yeah.
sometimes. MGH and some of those big ones they'll do that.
Yeah, that's right.
So
That's
you
right.
say here, you from this graph, you get knowledge graph, right?
Yeah.
So how do you get knowledge graph?
The knowledge graph this one is from a cell paper. They have analyzed which genes have different possibilities of copy number alteration. So these copy number alterations have been demonstrated to be associated with the drug response. So for example, if a gene are more likely to have all copy number alterations, they are more likely to present drug resistance.
But is it,
So-
I mean, it's manually constructed or it's automatically, like LLM can generate knowledge graph, right?
Yeah.
So is it manually constructed knowledge graph or the automatically?
You can interpret as generated by other people.
Right.
Yeah, we have used the output from a cell paper for this.
Hmm.
Yeah, that's how we proceed with this one. And we have developed this model and we have demonstrated some use case. We apply this model to multiple myeloma and we identify a specific resistant tumor cell state and which is more enriched in resistant patients and in advanced tumor stages, which indicate and demonstrate this tumor cell state is more likely to be a resistant tumor cell state. And we have done some downstream analysis like the differential gene expression and also the copy number alterations. And we have finally identified 3 biomarkers, which indicate because when we look into the clinical cohort and the final panel here is the clinical cohort, we look into the clinical cohort, we found these 3 biomarkers tend to be overexpressed and expressed after first line and second line treatment, which indicate that they maybe serve as the resistant biomarkers in the clinical patient. And that's the final discovery from this study. And on top of that, we also have got another paper just accepted in Nature Communications, and which also built on top of that work is we have developed a system to evaluate large language models for predicting drug response, because we have developed our own graph enhanced large language models, but which inspires us, what about the other large language models? There are so many models out there, how they perform in this context. So which inspires us want to evaluate how the existing large language models performs in this context. So we have developed this system, we have built the web design, we have built the pipeline, and also we have more curated data.
So which LLM are you using this? Which large language model?
We use we use, I think it's 6 or 7
DeepSeek,
large language, no, we cannot use DeepSeek.
so which one, Llama, Quinn?
We have Llama 3 and GPT-4. That we include as general purpose large language model. We also utilize the specialized large language models developed in this area. For example, there are many single cell related large language models developed. We evaluated all those relevant ones. Total, we evaluated 8 in the unified system, and we built this command line tool and interactive web server. And we also evaluated through the layer freezing or the fine-tuning strategies and based on our curated database to evaluate the model's performance in predicting drugs. And this is some examples of how we show the performance and general the conclusion. And also we have evaluated the GPT-4 and we add the GPT-5, GPT-4 and the other variant in the revision process. So in their like using the prompt to evaluating their drug response prediction. And the overall conclusion from this study is we have identified specific models such as SC Foundation and UCE. These 2 models performed quite well in predicting drug response. But the general purpose large language models doesn't perform well. So that's the primary
Like this
conclusion.
seems pretty interesting. So this work was done by you and your collaborator student, right?
Yeah.
These students are from which department? What major?
My students.
Hmm?
My students. The first author is actually he is a computer science master and involved in my PhD. But he has very, I think strong. Maybe I'm not sure compared with
Yeah.
students here, but he's pretty good at computing. So he is the first author, my PhD student. Yeah. And we have, this is the recent work we have done. And so there is take home message here is I want to demonstrate the graph AI and the multi-modal data is very useful. And we have proved that through our different research studies. And actually that is the first part. I just finished my first part. I have the second part is in the biomedical informatics. I would like to briefly go over so that you know I'm not only work on the single cell omics, I also work on different type of data, but I limited to by the time, so I will just give you a brief overview. We work on the image-based drug discovery as well because drug is my favorite topic. So we have utilized the multi-channel images purely based on the multi-channel images and to understand how the morphology changes respond to specific drugs. So that's the work also just accepted in the Nature Communications, and we identified the embeddings, the morphology changes after the cells being treated by drugs. So that's the goal of this work. And we have been using graph AI and multi-modal data in other contexts like the disease progression in Alzheimer's disease. We are not working on cancer purely. We work on different type of disease. And this is the Alzheimer's disease. We have using the medical image, MRI image, and genetic data to unveil the unified embeddings, which can help us to identify whether this individual is diseased, Alzheimer's disease, or it's normal patient, or it's the MCI, that is the cognitive deficient patient. And so we also did the interpretable analysis. We found that specific genetic features are associated with their brain images. And this indicate their disease possibility and likelihood. So which is very important. And we also showcase these features in the brain. The upper row shows the sagittal views of the brain and how those features are important into the for the prediction. And moreover, the last one, I think it is another work we have been doing the chronic kidney disease or another type of disease, but they are relevant because we use different type of data. Because why we work on the chronic kidney disease, because it's very heterogeneous. Different patients can progress differently. So that's why we work on this, and we have the cohort. And this is another study in Wake Forest when I was there. And we use the graph search and also we use the discriminative decision tree to identify the patient disease progression trajectory. And this is embeddings that we get to, which is very clear patterns, which demonstrate that the embeddings are very meaningful. And the interesting things here is we identify 2 trajectories. Where the patients have these 2 trajectories, we have the patients at a similar age. the red circle ones and the blue circle ones, they are at similar age, but their EGFR, that is indicate of the kidney function, are very different. We indicate this, these two circled trajectories represent different risk of kidney disease progression. And axis, which is that? It is just the embedded space. It doesn't have real meanings, embedded space. So we find that the green ones and the red ones are the patients at similar age, but very different kidney function. And we found that the risk, the white patients, I think it's white patients. Yeah, it is white patients. Yeah, it's blue ones, this one. Yeah, higher risk of kidney dysfunction. And also it's male. So our findings in this cohort indicate that the white male may more likely to have kidney dysfunction, even they are at a similar age with the other risk of cohort. So that's the major finding of this work. And on top of that, we have built an interface, which we call as the clinical decision tool, using this model, which that is Travis, which is an AI-driven visual clinical decision support tool that integrates multi-modal data, including the EHR, the lab test result diagnosis, and also demographics and other genomics data to model and visualize the patient-specific disease progression. So that's another work that we have demonstrated very clinical and translational impact. So I think the take-home message, again, is graph AI is important. Multi-modal data is important. It helps us to understand and to improve patient health, the human health. And and also it will contribute to the development of the future, maybe trustworthy, generalizability, and the robustness, which is the major topics that we all care about in the computer science department. So that is the take-home message here. And because of my research experience on different type of data, I would say there are a lot of collaborations that can happen within UCF with different departments and different investigators here. And I have actually I'm actively submitting HR1 and the other equivalent grants, and we have the R25 submitted as well. And we are also trying other foundation grants. So I would say there are a lot of opportunities to work together as a team and to submit the multi-PR grants. Last, I would like to acknowledge my collaborators from Wake Forest, Mayo Clinic, and University of Florida. And also, I'd like to thank my funding support from NIGMS and AI2 Heal Catalyst grant, and other support from the House Cancer Institute in University of Florida. Yeah, thank you. I hope I didn't take much.
You're well on time. So quick, maybe one or two questions from the audience, and then I have one question.